Welcome student. This is for AS Biology Lab. For this pandemic situation, we are unable to do the lab in the classroom. So that is why I am thinking that I'll do something for the student. That is why I'm here in my YouTube channel to solve some question, and I'll show you how to solve and everything you will able to learn. So that is why it will be very helpful for you. Okay, today we are going to solve the question number 2012 October November variant 3. This is AS Biology Lab. I'm Mr. Jalil Islam. Please subscribe my channel. Okay. Today, at first, I'm showing to how to solve it. Actually, you know that practical class is not able to do without actually through the video or something. We need to do it physically, but school is closed, so we are unable to go to school and to do physical class. So that is why I'm here. I'll show you how to do that microscope part of this question. You know that there will be two questions in the question paper in Cambridge AS level exam. So first one will be biochemicals and second one will be the microscopic. So for the microscopic question, we are doing today, October November 2012, variant 3.1. The slide, I'll select the slide and I'll set it in the microscope, and then I'll take the picture and then I'll solve it in the classroom. Okay. So for today, it is it is very difficult to understand that which slide was given in that particular question. But I usually used to see the mark scheme and question paper, and then I will try to understand that which slide will be suitable for this question, and then I will try to do that one. Okay. So I am giving you the question in this question, that is slide J1. Okay. So I am selecting a monocot root of this for this question paper, and that is maze root. At first I'll show you how I am setting the microscope. Okay, this is the slide and I'm showing you how to do it. Okay, so first of all I am doing the thing. Okay, so first of all I am doing this thing. I will start with this question. You know everything. You will be given only two microscopic objective lens, that is yellow one is 10 and yellow one, and another one is blue, that is 40. So as well as that, you will be given eyepiece lens, that is also, it will be written 10x. So for low power plant diagram, you will select the microscope. At first you will be given the slide beside the microscope, and then you will set the slide on the stage, and after that you will use that mirror to fix the mirror, and then you will use these two knobs. You see these two knobs, that is close focusing screw and fine focusing screw. Okay, so just it will move that this actually tube, as well as it will move the lenses, so you can see the microscope slide. At first you will set the light, that is why when you see the light you fix it the mirror, and this is the simple light microscope. So most of the time in the exam center will be given that. I have in the behind, I have another one that is actually higher one. Okay, you see, sometime in the some exam it can be given, but I am not using it. It is actually the same, but with the light source, as well as it is binocular. This one is monocular. Okay, so at first I will see here, and then I will move that cross focusing screw, and then I'll first set the fine focusing history, and then I'll set the microscope. Okay, this is for the setting of microscope. I'm not showing you. I have prepared another baby video for the setting the microscope, and I'll show you. And I have setting it. I have used that microscope. I have taken the picture with the microscopic camera, and then I am setting that question paper. I am showing you the picture of the Xeon slide with this question paper. Okay, so I am going to the going to solve that question. Okay, so I'm going to share that question first. Okay, okay, this is the question paper you are looking for. This question paper is October November 2012 variant 3.1. Okay, so I'm going to the question paper, the question number 2. At first I'll try to understand the question and then I'll to solve it with you. Okay, so from the page 8 the question 2 will question 2 is started. uh, at first I will read the question paper. Z1 is a slide of transverse section through a plant root. So they are saying that this is the transverse section of the plant root. Describe one observable feature, one Z1, which identifies the specimen as a root. Okay, so just you can say, sorry, you can say this is a root. How we can identify this is the root? You can identify root, that is you can see the slide and you can identify that how this is the slide is a transverse section of root. So you can say that lots of things you can identify. You can see the root hair cell, but most of the time you will not see when that slide will be cut very, but you can identify if there is actually endodermis present, if there is pericycle present, vascular tissue in center. So I am writing that by vascular bundle or vascular tissue in center. Just you will just write it and then you can answer this simple question. Okay, just you can write it. Okay, otherwise you can answer the vascular bundle in the center or endodermis present, uh, then pericycle present. Okay, so I'm going for the next part. Okay, that is for okay, question number A2. There is asking, draw a large plant diagram of a whole specimen on Z1. On your diagram, use a label line and label to show the cortex. Okay, so this is the plant diagram. So you will do this question. You will at first you will observe that slide and then you will plan yourself how to draw it. Planning means that you will spend at least 1 or 2 minutes to identify different tissue, and then you will plan yourself that how you can draw it. Okay, and then I am just saying that some common features of drawing, that you will use 80% of given space, you will use sharp pencil, you will not use any feathery line, any cutting line, any overlapping line. Your diagram should be proportional. Okay, as well as I am showing you that in the microscope, with the microscope, uh, you see there is actually with the eyepiece lens there is eyepiece graticles. So you can use that graticles and you can measure different layer of tissue and the size, and then you will draw the proportional diagram. Okay, so draw, you will here the space is given, so just this is the space. We'll use around 80% of this given space. Okay, I'm showing you the space. will use around 80% of this given space. Okay, so this space you must use for drawing purpose. Okay, so you must remember you must use 80% of this given space. Okay, after that I am showing you that the picture of J1, and then I am going to show you how to draw it. Okay, okay, so I'm starting the picture. This is the picture of J1. You are observing here the picture of slide J1. As you are seeing here, at first you need to identify there is epidermis and then cortex, then endodermis, pericycle, and vascular bundle in the center. Vascular bundle means there will be xylem and phloem, okay, and other tissue, other observable tissue. So at first I'll draw this diagram to identify the labeling, and I'll show you how to draw. I'll annotate with the picture, and then I'll show you how to draw it, and I'll show that I have drawn a picture for you, and then I'll show you. Okay, so for this drawing you need to draw a whole diagram, so that is why at first you will draw a circular line or circular layer of the epidermis. Okay, so this is the I'm drawing. You see, this is the red color pencil I'm using. I'm drawing that circular shape. This is for outer layer of epidermis. Okay, most of the time you'll try to draw the specimen as it is, but you will not able to draw any singular cell here, because this is we are drawing a plant diagram. Okay, and then just I'll draw another layer, that inner layer, inner line for showing the epidermis. Okay, so this is for epidermis, and then you will see this is endodermis. Okay, this is endodermis. I'll draw a line for endodermis here. You know, this is monocot root, so there will be another layer for, actually that is called pericycle, but I'm drawing endodermis first. Okay, then I'm drawing a pericycle. There is another layer of single cell that is called pericycle. So you will, when you will observe, you will observe it very okay. And then I'm going to show this is xylem, the red color cell, that is xylem. There is a lots of xylem, but you will not draw any singular cell. The big size cell that is you are observing, that is also xylem, but you must remember you will not draw any singular cell. Okay, then I'll just draw fewer of phloem. You see, this is the phloem, so you will just draw some phloem. Okay, so this is the phloem. Okay, this time I am not drawing all of them just for showing. I'm drawing fewer of them. This is the phloem. Okay. Okay, this is phloem. So you need to so I am using this, and now I am doing one thing. Just I will move the picture. That is why you can easily understand that how my drawing is looking. Okay, so just I'm moving. Okay, so I'm moving it. Now I am going to show you. Now I am going to show you different part of it, so you will able to understand. This is the cortex. Okay, so you need to label this part, and you already know this part is epidermis. Okay, this part is epidermis, then endodermis, then this is cortex, and this part is xylem, and I'm drawing some phloem. Okay, so I'm going to label it epidermis, then cortex, endodermis, okay, and xylem and phloem, it is in center. Okay, so I'm showing you this larger part is xylem, and this is the very smallest part, that is phloem. Okay, so this is annotation is only for that it will you will understand. This is the whispered, but you do not need to label it. just just you label the cortex. Okay, as it is asked in the question paper, I'm going to remove this picture, so you'll understand again. Okay, I'm going to show you how when I have drawn another picture with that actually pencil and paper, so I'm showing it again. Okay, okay, this is the picture I have drawn for you. This the picture will be look like it. So you see, as well as that I have given the picture beside it. That is why you can understand that the epidermal layer you see, this is for epidermis, and this is for cortex, endodermis, and then after that inner part is phloem, and this is part is showing xylem, and this is part is for showing phloem. So I'm showing you how to draw a plant diagram. You know, the plant diagram should be proportional, no cell should be drawn, as well as you cannot use any feathering line. You will find out some problem with my picture also, but it is almost, I think, okay. So if you can draw, I think you will be able to draw better than this picture. Okay, So I'm going the question paper again. Okay, so this is the question paper. For second draw part of this question, that is A3, I'm also reading. This time you need to draw a plan diagram before. This time you need to make a last drawing of one group of 4 complete touching xylem vessel as observed on the specimen J1. On your drawing, use a level line and level to show the on lumen. Annotate your drawing with one observable feature. Okay, so you must remember the mark given is, that is 5. You will draw 4 touching xylem vessel, as well as you will use a label line to label one lumen, and then you will annotate your drawing that how you identify this is xylem. Okay, so for this picture I am saying that you will use the microscope again, but this time you will move your microscope to 40, objective lens 40, and then you will use 10, that means 400 times larger. It is little bit difficult, so you need to practice more and more. That is why you can able to set the microscope properly. Okay, as well as I'm going to show you the picture. I'm going to share you the picture of from here, and then okay, this is the picture of center part. That is why I have given that actually larger picture, that you can easily understand that is actually 400 times magnified, so you can easily understand that question, as well as easily draw from here. Okay, so for drawing I will annotate how to draw, and then I'm drawing on thing that I will zoom it little bit larger. That is why you can identify. Okay, so at first you see how will identify the xylem. Xylem will be with actually stain red color, and then there will be large hollow lumen, and that cell wall will be little bit darker, as well as it will be little bit thin. Okay, thicker, sorry, thicker. So you will easily identify by observing. You see, this is the xylem. Okay, so this is the xylem, as well as beside it this is xylem. Okay, so you will just select on a large cell, and there will be some surrounding a small cell. You will just select one bigger cell, and then 3 smaller cell from here, and then just you will draw a large drawing. It is called detail diagram also. Okay, so that is why I'm going to show you how to draw it. Okay, just first if I select this cell, and then just I will draw this cell first. Okay, and then actually it is very difficult because it is annotated difficult. Okay, then I will just use another inner line to show the cell wall, and then just I will draw these 3 touching cell. Okay, just these 3 touching cell. And you must remember, for drawing the xylem touching xylem vessel, you will not use any intercellular space. Okay, so there is very little intercellular space, so please remember, but it is very difficult to draw without intercellular space. Just I am showing, I'll draw this 3 cell, and then I'll show. Okay, so these 3 cell, just you can draw from here. Okay, so as well as how to annotate, I will show you. For annotating, you will write down the something how you can identify the specimen, or how you can identify this is xylem bacil. Okay, so this is the drawing. This is the way you can draw. I am showing you. I have a draw uh, another picture, so you see. Okay, so you see I have drawn the cell, that is I have, this is this two line. Okay, so this is the, okay, so this is the picture of this uh, detail drawing. Okay, then you will draw a level line and to level lumen, and then you can label the thick cell wall, that is stain, as well as you can stain red, or you can write down that hollow, large hollow lumen with no nothing inside or something like that. So that is that will identify this is the xylem vessel. Okay, so I'm going to solve the second part of this question. Okay, I'm showing you. I'm going to, okay, this is some calculation they will ask always, that is actually called calibration or something there. I mean, I'm reading the question. Figure 2.0 show a diagram of a stage micrometer scale, and that is being used. Calibration of eyepiece reticles and division of eyepiece reticles, uh, on division on either stage of micrometer scale or eyepiece reticles is the distance between two adjacent lines. Okay, the length of one division of the on this stage micrometer is zero point 0.1 millimeter. So how you can identify? Okay, you know that stage micrometer will be magnified more, so you can identify easily the larger scale that is showing, that is actually that is stage micrometer. Okay, and the smaller one you are observing, that is eyepiece reticles unit. Okay, so from here you can identify, you see, you will count between 1 to 1. Okay, just I'm counting here to here, you will see this is little amount I'm counting here, and there will, you will identify, you will see that there is 10 division of eyepiece reticles unit. So 1 millimeter is equal to 10 division of eyepiece reticles unit. You must remember this thing. I'm going to then calculate it. Okay, just, okay, so I'm going to calculate it here. Okay, so 0.1 millimeter will be divided by 10. Okay, divided by 10, and that time the answer will be 0.01 millimeter. Okay, then you need to convert it to that actually um, micrometer. Okay, millimeter to, then we are going to convert it 0.01, sorry, 0.01 millimeter. It will be multiplied by 1000. You already know micrometer. Okay, so I'm going to convert it to micrometer. It is very difficult to annotate. Okay, so it will be 10 micrometer. Okay, so after that we are going to calculate and count to show uh, uh, the size of that cortex. Okay, So for this you will be given for 3 marks. I'm going to next. You see, this is that picture they're given. The figure 2.3 is a photomicrograph of showing the part of an organ from a plant of different species. So figure 3, figure 2.3 shows a photomicrograph taken using the same microscope with the same lens as figure 2.1. Use a calibration of eyepiece Gattacus unit from B1 and figure 2.3 to calculate the actual length of the plant tissue from X to Y. You will also lose mark if you do not show all steps of calibration and do not use appropriate unit. Okay, so you will be 2 marks will be awarded. You see, uh, from here to here they have asked y to z. Okay, y to x to y to x. Okay, that means endodermis to epidermis. You will not endodermis, actually this is actually stem. So this is the part of cortex. You will just count it. You will count from here to here, and I have counted it. There is actually 24 eyepiece Gattacus unit. Okay, so after that you will just write it here. You will write down x2 y is equal to 24 unit. Okay, 24 eyepiece Gattacus unit, as well as you will multiply it with 10 micrometer. Okay, and then you will write down the answer. So the answer will be is equal to 240 micrometer. Okay, this is the answer. For just this work you will be awarded 2 marks. Okay, and I'm going for the next part. Just I'm will be little bit hurry. Okay, for the next part you will that same picture 2.3 you will observe, as well as that you will prepare a space below so that it is suitable for you to record observer of difference between the specimen on the slide Z1 and the figure 2.3 to include the vascular tissue and at least 2 other tissues. Okay, so this question is very little bit tricky, so you must remember, uh, that is why at first you will be there is actually I'll see the mark, there is 5 marks, so you need to draw a table and with the headings, that is and then you will write down at least 4 differences, but you must write down the differences, one from vascular tissue, at least one from vascular tissue, at least 2 from that other tissue. Okay, so this is the last question, and I'm showing you how to do that. Okay, okay, this is the 2 picture for your observation. I have taken these 2 picture beside, that is why you can easily observe as well as that identify the differences. Okay, first you see There is actually cortex that is very smaller. This is the cortex. It is very little bit larger, where the cortex is smaller here, as well as that you see that epidermis, as well as the epidermis, endodermis is not present here, as well as the position of the vascular bundle. It is actually in the center. Number of xylem vessel. Okay, so everything you are observing here, the phloem, cambium, everything. Okay, so I'm showing you the picture. Then I'm going to talking about with that actually the differences. Okay, so this is the differences I solved it before. At first you will prepare the table like that. You will write down that feature. You must remember, you will write the feature. Okay, you will write the feature first, and then you will write down the Z1 and figure 2.2. Okay, after that, if you talk about vascular tissue or xylem position, you can write down in the Z1 that in the center or inner or near to the epidermis. Pericycle or phloem tissue surround the xylem. Okay, for figure 2.3, you can write down that near to S or in epidermis or phloem tissue outside the xylem. Okay, quantity of xylem or vascular bundle you can write down that is very fewer in J1, as well as then you can write down more in actually figure 2.3. And then quantity of quantity of vascular tissue you can write down. I already have discussed endodermis or pericycle or stele. You can write down in Z1 it is present, has or yes. In figure 2.3 there is no endodermis pericycle, so you will write down the absent or not here. As well as epidermis, you can write down the thicker, wider. Okay, whereas it is thinner in figure 2.3. Parenchyma tissue you can write down absent, or you can use a cross sign to show it there is no parenchyma. But in figure 2.3 you can write down it is present, our check mark. Okay, and cortex you can write down that there is a larger cortex in J1, whereas a very small cortex in figure 2.2. Okay, you can write down narrow or thin or fewer. Okay, this is the way we will write it. So if you can write correct 4 differences, at least 2 from other observable tissue and 2 on from vascular, 1 or more from the vascular bundle, so you will be awarded 5 minutes. This is that all about this question. I'll come with you with I'll solve here another question, and as well I'll try to solve with that biochemicals also. Stay with us. I am trying to do something for you. Okay, so please subscribe my channel. That is why uh, I can come again and again. Okay, so thank you, student. I'll Allah Hafiz.